Okay. All right, hello. Today I'll be presenting on microbes and the environment. Sorry about them. From protists to humans, all animals and plants are inhabited by microorganisms. Microbes benefit their host on a range of things, including metabolic, immunological, and behavioral avenues, but there is still a lot that we do not know about interspecies interactions. As we know, microbes have evolved to adapt to extreme environments such as deserts, intertidal zones, hydrothermal vents, and oligotrophic seas, which have low oxygen and low nutrient levels. Some microbes' motility allows them to wander through their environment to find nutrient sources or to escape unfavorable environments. However, some microbes are non-motile and are restricted to their local habitat. There have been reports that non-motile microbes can use the motility of other microbes in their vicinity, which is called hitchhiking. Hitchhiking has been observed with both prokaryotic and eukaryotic microbes, and many of them are pathogenic to humans or plants. Currently, research is being done on how this can affect human and plant health, and a new subfield of microbiology may occur. Also, advances in DNA sequencing have shown us that host-microbe interactions are situation-dependent and dynamic. Research is being done on manipulating microbiomes in zebrafish models to see how it affects their pathogenicity. Microorganisms are extremely important in plant development, nutrition, and resistance to stress. The structure of plant microbiota is considered host-driven, especially in the rhizosphere, which is the area containing the roots and soil around the roots. When the microbiota is described as host-driven, it means that the microbial communities are shaped by diverse rhizodeposits or metabolites. It is thought that plants and microorganisms co-shape plant microbiota as well. Through coevolution, plants and microbes have developed communication channels, and research proposes that small RNAs are necessary mediators in plant-microbe interactions, and that they aid microbes in shaping the rhizosphere. Also, microbes like fungi, bacteria, and viruses living within flowers are thought to affect pollination. There is evidence that microbes influence pollinator attraction, but bumblebees and nectar yeasts are considered the most prominent attractors for pollinators rather than the other microbes right now. This is still majorly understudied, but future research will be conducted where microbe diversity and pollinator processes are researched. Additionally, plants release metabolites from their roots, which are used as growth substrates for certain microbial strains. They can also perform as signals in mediating plant-microbe interactions. Flavonoids and strigolactones are some of the important metabolites in aiding microbial plant symbiosis. Learning how these interactions work could lead to discoveries that will aid with agriculture. The study of Sclerotinia sclerotiorum, a problematic soil-borne pathogen of many important crops, will aid humanity in understanding crop pathogens in soil as well as how to maintain beneficial soil conditions for a healthy microbiome soil community that mitigates soil-borne diseases on its own. Farmers are beginning to heavily consider probiotics, which are live microorganisms that could possibly fulfill the roles that antibiotics have. Research suggests that probiotics can help chickens, pigs, and cows put on weight faster, digest food better, and withstand pathogens better. But farmers already have many ways to raise healthy livestock without relying on antibiotics and probiotics, like adding enzymes to feed, choosing different animal breeds, and putting in new infection preventatives. Most of them are skeptical that microbes would be worth the price, though. Moving on from livestock, the human body has its human microbiota, which can be found on the skin, in the nose, in the mouth, in the gut, and basically everywhere. And as we know, we gain these bacteria right at birth and in the first years of our lives. It is said that each human body hosts 10 microorganisms for every human cell. Microbes contribute to digestion, vitamin K production, development of the immune system, and chemical detoxification. And of course, microbes are necessary for making the foods that we so enjoy, like bread, cheese, and wine. But obviously, microbes can hurt us too, as we've been seeing this semester. Humans are having trouble feeding the growing population with the limited land we have, and with dwindling freshwater resources on top of global warming, new solutions are needed soon. Research is being done on how we can use microbes to capture CO2 from the atmosphere and convert it to something else. Research is also being done on how probiotics could help us with SARS-CoV-2 through gut immunity. Currently, microbes are used to clean up pollution in processes known as bioremediation. Bioremediation uses microorganisms to reduce pollution through the biological degradation of pollutants into non-toxic substances. This ability results from the fact that microorganisms exhibit a wide metabolic diversity and thus are able to degrade an equally wide variety of chemical compounds. This relates to what I said before about the CO2. However, since many pollutants are cellular poisons, it is not surprising to find that they also inhibit microbial growth and activity. Microorganism activity is responsible for the major cycling of elements in the environment, so any impairment of microbes will have negative effects on nutrient cycling. Studies on perchlorate, which is used in rocket fuels and munitions, have been shown to impair thyroid function. Studies show that we can use perchlorate-degrading bacteria to remediate it. The research has been using bioreactors to decontaminate perchlorate-filled waters. Bacteria are being studied everywhere to aid in diminishing pollution's effects, but microbes can also be causing pollutions, like cholera, which lives in the water. Studies are currently underway on microbes with the goal of planetary protection as well as the search for life. The responses of microorganisms to certain space factors like microgravity, cosmic radiation, solar UV radiation, and the space vacuum are also being studied. NASA started a program called Genes in Space 3, where they had astronauts sequence the DNA of microbes found aboard the International Space Station. The successful sequencing and microbe identification marked the first time unknown organisms were sequenced and identified entirely in space. Mangrove forests have some of the most productive ecosystems in the world, and it is due to the unique environment and its microbes that provide nutrients to a wide range of organisms in its ecosystem. It has been found that major contributors to the nitrogen, sulfur, phosphorus, and carbon cycles include mangrove-associated soil bacteria. Black mangrove root systems are bracketed with soil phosphate solubilizing bacteria that aid in the oxic and anoxic recycling of carbon. Mangrove litter decomposition and the conservation of nutrients are headed by fungi microbes. A trichoderma fungi species connected to the mangroves is a contributor to the solubilization of soil phosphate and the improvement of the mangrove species Avicennia marina. Actinobacteria are known to also aid in mangrove litter decomposition. Yeasts, marine cyanobacteria, lactobacilli, and saprophytic fungi are associated with mangrove ecosystem health as well. Studies displayed that microbial counts became reduced with the increasing soil depth. The bacterial consortia on the tongue form the structure of a patch mosaic. Keratinized epithelial cells are the center that the consortia is organized around. Oral microbes reside in site-specific areas and do not appear ubiquitous, species-wise, in relation to location sectors. It is theorized the oral microbes replicate by clonal expansion as they appear to be 10 layers thick over the oral cells. In the studies, the most abundant microbes were Rothia, Streptococcus mitis, Veillonella, Streptococcus salivarius, Actinomyces, and Necessariaca. Venom is known to be a producer of natural antimicrobial products. This led to the misconception that venom and venom-producing glands are sterile environments, but research has shown they have microbes of their own, although of which is currently unknown. The Initiative for Venom-Associated Microbes and Parasites, IVAMP, are heading a worldwide research project to begin looking into the venom microbiome. Thank you for watching. If you have any questions, just email me.